This is the Kratom Science Journal Club with Dr. Jonathan Cachat, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. I have an announcement to make. Next week, we will be hosting a Reddit AMA, that stands for Ask Me Anything, with our own Dr. Jonathan Cachat on r/Kratom. That's reddit.com/r/Kratom. In addition to the Kratom stuff you hear on the Journal Club, Dr. Cachat is a neuroscientist who studied pretty much all DEA Schedule 1 to 4 drugs as part of his graduate work, and he knows a lot about cannabis indoor growing and helped found the Cannabis Museum in Ohio. Look for the announcement for the AMA. That will be next week on reddit.com/r/Kratom. The journal article is titled Investigation of the Adrenergic and Opioid Binding Affinities, Metabolic Stability, Plasma Protein Binding Properties, and Functional Effects of Selected Indole-Based Kratom Alkaloids. And I think I pronounced everything right so far, but that's going to end pretty soon. And this was in Journal of Medicinal Chemistry. We have Dr. McCurdy again on this. A lot of people from College of Pharmacy at University of Florida. And they're basically studying how a lot of the alkaloids bind to different opioid and adrenergic receptors. And so there's some of this study I understood, and then there's like a middle part where I was like, okay, I'm just going to have to ask what the hell's going on here. But
it certainly is like an alphabet soup of receptors, and then they get into like specific proteins or components of those proteins and the receptor molecules. So there's some real detailed like analytical chemistry at a molecular level going on here. But I think you summarized their goal pretty well.
And I was just going to go over again, just for the listeners now, opioid receptors. I think most people know basically what they are. And adrenergic receptors. Now, would that be more, since a lot of these alkaloids bind to the adrenergic receptors, would that be more of maybe the stimulant properties of kratom, and the opioid receptor binding would be, in general, the sedative properties of kratom and pain relieving?
Yeah, so more or less. I mean, there's lots of crosstalk between these two systems and overlap because they can bind the same ligands. So like what we would typically think of as an opiate agonist can also bind in like alpha-1 or alpha-2. They affect the same downstream messenger systems. There's actual instances too where like, the mu-opioid receptor will form a dimer with one of the adrenergic receptors, say like alpha-1. So the two different receptors of two different systems can come together and form what's a heterodimer, and that changes how the information is relayed down into the cell. Like you said, with the opiate system, there's 3 G-coupled protein receptors. We've talked about them a lot, the mu, delta, and the kappa. Mu is generally responsible for the therapeutic and like pain relieving effects, and also the rewarding, pleasurable effects that can then, are currently theorized to be behind addiction. When it comes to the adrenergic system, it's definitely a little more complicated. But what is typically described or summarized conceptually is that the adrenergic system is what is behind your fight or flight response, or your rest and digest response. And so that's the back and forth between the sympathetic nervous system and the parasympathetic nervous system. For our purposes in this study that we're looking at now, it's the sympathetic nervous system, or that fight or flight mode, that the 4 adrenergic receptors, alpha-1, alpha-2, beta-1, and beta-2, are focused in this paper. So what I think the major takeaway home is just that when alpha 1 receptors are activated by the sympathetic nervous system, your pulse goes up, your blood pressure goes up, your pupils dilate, you sort of spend less time on metabolizing food within your digestive system. So that's sort of like getting you ready to fight or run. And the opposite is true with alpha 2. The alpha 2 receptor, when that gets activated, it reduces your heart rate, it reduces respiration, it reduces your blood pressure, it then reduces like inhibitions on your GI tract. So you're sort of, coming down from this state of wild anxiety.
Yeah, and now we're talking about how different kratom alkaloids bind to these receptors. We talk about the ones that are the two most famous ones, the Mick Jagger and Keith Richards of kratom alkaloids,
Exactly.
mitragynine and 7-hydroxymitragynine, and we can call them MG and 7O. And since 7O, they say it has the more opioid like effects, that would be the Keith Richards.
Okay.
But the other ones, I don't know. That's stupid. Whatever. But the
We might get mixed up.
so the MG and 7O, those are the two main ones. The other ones they're looking at here are corinanthidine, and we can call him Corey.
Corey, I like that.
Okay, 9-hydroxycorinanthidine, and we call that 9-Corey. Speciosiliatine, which isn't. We did one on speciophyline, which I decided was pronounced like that. And we call that speech. And there's another thing that they talk about, which I don't know exactly what it is. It's yohimbine or yoimbine.
Yeah.
What is that? Is that an alkaloid?
It's an alkaloid, a well-characterized alkaloid that has been in psychopharmacology for a while now. It's a sort of a standard opiate receptor binding ligand. Can I say a little bit on Corey?
Yeah.
So, the way that these authors set this paper up, they're essentially saying, the classic, well, there's a lack of scientific information. People could be getting hurt or people are getting hurt. We need more information. The part that bothers me is like, there's a lack of scientific information to provide guidance on this. So let's just ban it. They admit to it, so they go to ban, and that's the default, and where the politicians and the regulations come. But where they're getting at is we do need to understand information, and especially when it comes to good manufacturing practices, GMPs that the AKA is reporting. The purpose of the study, though, was to follow up on a finding, and I'll just read the quote here. Interestingly, it has been shown that Corey, a minor kratom alkaloid, acts as a functional antagonist at the mu-opioid receptor and can reverse morphine-induced inhibition of twitch contraction from the guinea pig ileum. Basically, what they're saying here is that it can replace morphine that's inhibiting these smooth muscles, which is what causes constipation. But it doesn't seem to result in a reduction of the analgesic effects as much. So they're saying like Corey in particular seems to be reducing the amount of contractions, maybe reducing the amount of pain effects felt, but it's more active, it seems to be that it's active at another receptor type different from the mu-opiate receptor. I think that's sort of the insight they keyed in on. And I guess another way to consider it is, well, if you can make a prescription drug that gives patients pain relief but does not give them constipation, maybe there's some targets there. Maybe with Corey, maybe if we learn more about Corey, we can figure out what makes it unique enough to get to that prescription, that type of prescription. Does that make any sense?
Yeah, and I even pulled that quote, and I was going to ask you about it too. And another thing is what we were talking about. We talked about it, I think, last time and a few times before. Was this entourage effect of alkaloids? And just another quote from the article I pulled. Whereas, we were talking about how if you isolated mitragynine and took that, it wouldn't have the same effect as kratom because there's a lot of
Mm-hmm.
different alkaloids acting, and that's basically what this whole paper is looking at. How the entourage effect of kratom works. And what they say here, it's important to realize that these alkaloids have been and are being investigated as purified individual entities. As such, the resultant data are not directly correlative to the complex plant mixture where they occur in varying concentrations or ratios that could impact each individual alkaloid's pharmacokinetics and pharmacodynamics.
Very nice. Yes, I have that highlighted as well, and that is very.
Yeah.
I'm glad. I'm glad that you focused on that because I think it was just the last Journal Club um, uh, episode that we put out where we were talking about this potential entourage effect of the alkaloids in kratom, and so yeah, it's important to know that, particularly when you're trying to embark on these types of studies.
Go through like what they actually did in this study. I'm under the results and discussion section. It says that alkaloids were screened for their ability to displace bound radioligands at delta kappa mu and nociception opioid receptors, which they call DOP, COP, MOP, and NOP. They sound like the Seven Dwarves or something. And
Yeah.
at the adrenergic receptors. Um, First of all, what's a radio ligand?
So the analogy of a lock and a key is best used to when we're talking about the receptor binding. The key in this case is the ligand. It's the part that binds to or goes into the lock. And so basically, what they're doing in these studies is you have your lock, the mu-opioid receptor, and you have your key, the ligand that we want to bind to this receptor. The radio tag, you can think about it like putting a little tag on this key that glows, and it glows in very specific things. Like maybe it only glows when it's binded. Maybe it only glows after if it's not bound. But the radio tag is like using radiography or X-ray type stuff. It's a way to move and visualize and count the amount of keys that are in the lock or not in the lock at any given time.
Okay, that makes sense. Now, screen for their ability to displace bound radio ligands. What does that mean to displace them? Would that just
Yeah.
mean putting the key into different locks, or
Well, so I think the best way to think about it is naloxone. So if you're in the middle of a heroin overdose, the molecule, which would be the key, is stuck in the lock, and you could potentially die from that. When you give them that Narcan or that naloxone shot, the Narcan goes in there and rips the opiate molecule out of the lock. It displaces it from the lock. Um, so that's ligand displacement. Why they're doing it in this study is more related to how are they determining what binding affinities are. And so there's um, it can I don't know how many sort of um, physical or chemical psychologists or neuroscience we have on as listeners. So I'm going to just try to I'm going to stay sort of very high level. Um, you have to know that a binding affinity, which is either marked as Ki or Kd, the stronger there is binding on the receptor, the lower the Ki or Kd is. So a low number of affinity is stronger binding at the receptor. The way that you actually get the K number, the affinity number, is by looking over time, how long does it take your target molecule to displace 50% of the receptor binding in the experiment? So people talk about LD50 or
mm-hmm.
therapeutic dose 50. This is sort of the same thing. You're calculating the affinity based on timing how long it takes for 50% of the tagged compound to throw them off of the receptor. Does that make sense? Is that where your question was?
Yeah, yeah. I was just trying to get a general idea. And what did they actually do? I mean, there were rat tests and stuff, I think, in this study, but what are they actually doing to look at the? Are they just looking at the effect on mice or rats in here, or are they looking at everything under a microscope, or maybe like liver specimens or something like that?
There's a few different ways, and you're right. The study did have behavioral endpoints at the end. For the
Yeah,
people that are doing these radioligand binding studies, which I think we might have gone over a little bit in a previous episode, but what they're actually doing is they're getting a piece of, like a skin membrane, or it could be just a single cell, a single neuron. They're making it, injecting vectors that will increase the number of, say, mu-opiate receptors on that cell's surface. And then they can use electrical diodes to see when they get active, if the electrical signal goes up or down, just to make sure that it's efficient. And so they can both use cell physiology to look at the amount of flow coming in and out of these receptors, as well as the radioligands visually to count to see at what point 50% is reached. Is that clearer? It's not a live animal, it's a cell tissue culture,
hmm
that they're hooking electrodes up to and then taking pictures of.
I'll just read something I pulled out, and then it's kind of an introduction for a question I had. But the affinity
Mm-hmm.
of mitragynine was determined at all the receptors as a reference to compare the other kratom alkaloids, except at DOP and NOP, delta and nociceptive opioid receptors, where it had poor binding at 10,000 nm. So I'm kind of just looking at these tables, table 1, table 2, table 3, and it shows a list of the receptors. And this is an open access article, so anybody can look at it, and I'll have a link to it in the description. And then it has all list of all the alkaloids, and it has NM. What is NM as a measure of?
It's nanomolar, so it's a
Okay,
measure of concentration.
okay. And so, what do you? I mean, this table shows that there's different rates of concentration at different receptors. Um, so, you just have any
So
comment on the table or what that
yeah,
shows
so
us?
looking at table one, let's look at like the mu-opiate receptor. So the binding site is mu-opiate. It's the second one up from the bottom. Before they added the 7-hydroxy or the mitragynine, before they added the alkaloids to the cell tissue culture, the cell tissue was already exposed to an opiate ligand, so one that would bind to those receptors that was tagged with the fluorescent tag. Because this is in percentage, 7-hydroxymitragynine had almost 100.7% and 81.8% had displaced the radioligand the most, meaning that 7-hydroxy had a stronger binding affinity for the mu-opiate receptor, or stronger at least, than the bound radioligand. Um, all of the results in table one were just to essentially screen the alkaloids that they were looking at. So 1, 2, 3, 4, 5. They screened them to then say, which ones should we proceed with? And what studies do we need to go from there? So the strongest opioids are the strongest, the ones that displaced the radioligands the most, then went over into table two. And so in table two, we're looking at the kratom alkaloids, specifically binding to the opiate receptors. And so you can see that for the mu-opiate receptor, this is the third column, MOP. The strongest was the 7-hydroxymitragynine. Number 2 was the Specio one. Number 3, the 9-hydroxychlorothalonidine. Number 4 was the chlorothalonidine, and then number 5 is the mitragynine. And that's just looking at lowest to biggest in terms of the strongest binding affinity. So that's the order at
Okay.
that receptor that they fit. 7-hydroxymitragynine was the strongest, had the strongest affinity for the mu, kappa, and delta across the board. But that's not really the case for the other kratom alkaloids that we're looking at.
Yeah.
And so table 2 is what are the binding affinities for opioid receptors, and then table 3, what are the binding receptors specifically for the adrenergic subtypes. Corinanthidine, with the 41.7 is the strongest of the kratom alkaloids. It binds the strongest to this alpha 1D subtype receptor.
I mean, did they do anything to manipulate the particular alkaloids? Because there's, I'll just read this quote I pulled out. The binding data obtained at the opioid and adrenergic receptors show that the removal of indole methoxymoiety on Mg does not influence the binding affinity to the MOP, mu-opioid receptor. However, removal of the methoxy group results in significant reduction in binding to the kappa opioid receptor. And are they like manipulating? I don't even know what methoxy is. And are they the ones that are removing it, or is it just something that happens in binding?
Well, so you see figures like 2 and 3 have these 3D color chemical structures.
Yeah.
So what they're trying to do is essentially would say, if you have the key analogy, now we're talking about the bumps on the keys. And they're in the computer, they're changing the bumps on the keys to see what bump is most important in order to make the lock open and turn. So they're not actually modifying the entire molecule. They're like adding a hydroxy group would be to pop off a hydrogen and then add an oxygen and a hydrogen together. So they're modifying very particular attachment groups at a very particular spot on the ligand or the key, and they're seeing if that changes the conformation of the binding to the receptor. Does that clear it up a little bit?
Yeah, definitely. And they even said, and you said like with the oxygen, they even said introduction of a hydroxy group at position 7 and 7-hydroxymitragynine significantly increased binding to the mu-opioid receptor compared to the kappa. So I guess they're adding and taking away
the hydroxy group. So the chemical isn't changing. And let's just use mitragynine because it's easier for us to say.
Yeah.
They're not changing the chemical away from mitragynine. They're just changing like these secondary groups around the structure, the conformation structure.
Okay.
I mean, another way to think of it too is fentanyl is so potent because it has like 1 or 2 OH groups in a different spot than hydrocodone or oxycodone. And that change to the bumps on the key makes it so much, the fit so much better in the lock that it makes it very deadly. So these small changes, like it's called understanding receptor dynamics. So the ligand or the molecule that's coming into the receptor, the 4 points that it binds to the like cup on the receptor lead to changes, and then the receptor starts to twist. Well, you can remove, one or two of those groups from the signaling molecule, and it will still bind to the receptor, but it won't bind as strong, and it won't cause the turn. So they're like specifically going through one by one to say, okay, we know this part causes the turn. We know this part causes it to open up the channel a little bit more. What if we were able to change 2 or 3 parts of those? How would it affect what happens in the receptor? So it's very deep into the relationship between the ligand or the key and the lock, and to the point where you're looking at the bumps on the key and you're looking inside the lock to see how the binding actually occurs and how strongly it occurs.
And incidentally, I think that's kind of how synthetic drugs work, because I know my brother's a bodybuilder, and he was, a few years ago, he was taking this stuff that he said it was like a molecule away from methamphetamine, but it was technically legal because it wasn't methamphetamine.
It's how people get ideas for research chemicals or other synthetic drugs.
Yeah.
I can modify certain aspects of the drug and it will still bind to that same receptor that the drug of desire does, but the molecule's changed enough that it's not technically the same. Yes, it's where you would get like it's where you would get the prospect list. Like, well, if compound A is illegal, we can make B through Z slightly different enough, and then figure out which one binds to the receptor to get the effect we're looking for.
Are there a lot of studies like this with the kratom alkaloids? Because they're looking at them so closely that pretty soon, the scientists are going to be able to say, this is what this one does, this is how this interacts with the other one. I don't know how much of that's out there.
Yes, you're right in that not a lot of studies like this are being done. And so a lot of the studies that we have spoken about in the past are behavioral studies, and then they'll use a note like they'll either pull away the mu-opioid receptor altogether, or they'll see if it can be stronger than naloxone. But they're really just changing the drugs they're giving the mice and whether or not the receptors they're interested in are there or not. In this study, they're zooming in much closer and looking at, like I said, the bumps on the key and the bumps inside the lock in those 3D chemical representations to figure out specifically what parts of these molecules lead to the differences in the binding receptors. Not a broadly like macro picture of, is this receptor needed for this ligand, or is this, and how powerful is this ligand? No, there have not been very much that zoom into that level of analysis of like the physical dynamics between the receptor and the ligand.
And under functional effects, I pulled a couple things that were pretty cool. I thought it says, interestingly, A1 adrenergic antagonists have been shown to be effective in reversing the rigidity of the diaphragm, chest wall, and upper airway, wooden chest syndrome, produced by fentanyl, which suggests that mitragynine may be useful in curbing fentanyl-related overdoses. Would this be anything like how naloxone works?
I would say, broadly speaking, yes, because it's discussing how the interaction of two compounds or a slightly different compound with the same targets lead to different physiological results. But I don't know exactly. I don't think that they were suggesting that, like, if someone's taken fentanyl, we could use mitragynine to counteract the effects of the fentanyl.
Okay. And another one I was looking at under functional effects is speciosiliatine. Had anti-nociceptive effects at 10 milligrams per kilogram, which is pain blocking. However, this dose produced lethality in 5 out of 21 rats tested, indicating a narrow therapeutic window. So they found the LD 25, I guess, of species. So that's kind of another example why kratom works better in the entourage effect. Instead of, you wouldn't want to extract that alkaloid and take very much of it.
Right. Yeah. It's because it's found in low levels in the naturally occurring leaf. Mm-hmm. Yeah. You're not at risk for running into those issues. Wow. Yeah. And I think the other point too worth mentioning here, so they did the behavioral studies and like the tail flick tests, but they also were looking at how quickly the different alkaloids are metabolized in liver enzymes. And so they Cori, for example, is half of it is metabolized by your liver enzymes within 6 minutes. And so it's completely metabolized in 12 minutes. At least in these studies that are like, they take liver enzymes, they put them in a test tube, then they inject the compound of interest. But untangling this a little bit more, if we were to like whiteboard this out and add all of the alkaloids, we could then say, we're trying to figure out the entourage effect. Well, this one binds strongly to the mu-opioid receptor, but it's metabolized in the liver within 12 minutes. So the effects that we're seeing probably aren't related to this alkaloid. That's some of the stuff that this paper, the data that this paper is providing,
Mm-hmm.
that ends up being useful.
Part of the title is like metabolism. The plasma and microsomal protein binding corrected hepatic ratio suggests that
Mhm.
the alkaloids have low hepatic extraction ratio. So what does that suggest?
Cori was found to be unstable in human liver microsomes. So those are just enzymes in the human liver. Unstable meaning that cori doesn't stick around for very long.
It just says in general the alkaloids have low hepatic extraction ratios. Does that mean they take a while to get out of the liver? I mean, that could be why they work.
So I read that sentence to say that they are not degraded very quickly in the enzymes.
Okay.
In general, the binding correction and clearance prediction resulted in under prediction of clearance. It's important to perform in vivo in animal models to establish a correlation to better predict the haptic clearance of these alkaloids. Yeah, yeah, and frankly, I haven't come across any studies that are looking at those pharmacokinetic patterns, like how fast are these compounds or the alkaloids metabolized and then excreted. This is one of the first that gets into that level of detail. And I got to say, I think I'm with you on this one, and that the amount of detail and nuance, how deep this paper goes, I feel a little bit like a fish out of water to a certain extent.
Hey, you and me both. Welcome to land.
For sure. This paper, and one of the reasons why we picked it, is because we get a lot of questions about how the different kratom alkaloids affect the effects that you feel. And we always talk about the opiate the signaling system, and the adrenergic system comes into play sometimes. I think this is the first paper that clearly demonstrates that some of the alkaloids, like mitragynine and 7-hydroxymitragynine, bind strongly to opiate receptors, and some of the alkaloids in kratom do not. This paper then says, but the alkaloids that aren't binding to the opiate receptors are binding to the noradrenergic system and the noradrenergic receptors. And what this can sort of mean, and in the detail they're going in, like, it could lead to better treatments, it can lead to better pain medication that doesn't have the negative side effects, but it also can lead to better treatments of people trying to kick opiate use disorder. So like if you were to take cori, it doesn't bind strongly to the opiates, but it does bind strongly in the noradrenergic system, and in particular can lead to the one where you're decreasing your heart rate, you're decreasing your blood pressure. So you're basically like pushing the body into the rest and digest mode. So if you could make a new opiate use agonist at this alpha receptor 2 in the adrenergic system, you're essentially reducing the anxiety, or that anxious energy that you get in the middle of a withdrawal phase. And so I think that's a cool contribution that this paper makes. And then more broadly speaking, you and me are sort of discussing and navigating this science as it comes out in real time. So it's cool that we're getting to the depths of levels where I'm starting to feel lost, but we're getting to the depths of levels where the people that are doing these type of drug discovery and drug development research are now getting data points, particularly the ones when you're looking at that 3D representation of the alkaloids, 3D chemical representation of the opiate alkaloids. Understanding what makes the aspects of the kratom molecules bind to opiate receptors in a particular way is important, not only to determine safety, but it's also important to prove that maybe the dangers aren't there, and maybe you can learn from this and then change current opiate medications to make better ones.
And it also kind of confirms the final sentence of the conclusion that says the polypharmacology exhibited by the kratom alkaloids may support the claims made by patients taking kratom for the self-management of numerous diseases such as pain, opioid use disorder, and opioid withdrawal. So it's kind of even with all this complicated chemistry they're doing, they even say, well, basically what people have been saying is right. So it
Yeah,
kind of
well,
backs up the social science of it.
and it would suggest that just taking mitragynine alone for opiate withdrawal or opiate use disorder wouldn't be enough. Corey adds another piece to this puzzle to say it's slowing down your fight or flight response and turning up your rest and digest response, which is really what people need when they're trying to transition off of opiates and get rid of that. Like, I've heard people report it as like, I always feel like I'm like anxious, like right before they're about to take off from the starting block of a race. That sort of shaky energy that you have is your your sympathetic nervous system activating. If you could reduce the amount of activation, or as it seems Corey does, then there's an additional support for its use in opiate use disorder. So not just as not as potent, not as strong opiate, but also as the adrenergics, modulating the adrenergic system to reduce these effects.
Listen to me. Listen to me. There was a comment on YouTube on the last episode. So this guy, JM, commented on YouTube about the kratom cultivation study, the greenhouse study. He said, I think this study is rather short-sighted, valuable, yes, but in terms of studying the chemotypic profile, looking at alkaloids only is likely missing the full picture. I personally believe the alkaloids are only partly responsible for the bioactivity this plant provides. Focusing on alkaloids as a major component of bioactivity is an old approach and has allowed active components like salvinorin to be missed for years by Hoffman, who was convinced it had to be an alkaloid. Mitragynine is bioactive, no doubt, but there's more to this than just the alkaloids.
Yeah, so salvinorin is a terpenoid, and mitragynine is an alkaloid. Yeah. So, I gotta, thanks for the comment, JM. And I gotta say, good job on noting the difference between alkaloid and the terpenoids with salvinorin. So I would say he's right in that if you're just looking for nitrogen-based alkaloids in a plant to understand what its effects are, and you're not also looking at the terpenoids, you would miss something like salvinorin. But I would also just say, caution that mitragynine and 7-hydroxymitragynine are like really the only things we know about. I don't know what terpenoids may be in kratom that we would want to look at. And you got to start somewhere, really. So like the study can't be short-sighted because it's the first one. You got to start somewhere. But he's certainly correct in to say the chemotypic profile and the biosynthesis of terpenoids and the alkaloids is important and responsible for the bioactivity that the plant provides. Maybe he knows of some sequester terpenoids like salvarin that are in kratom. I don't know if we're aware of any terpenoids or diterpenoids that are in kratom that have binding activity at this point. But it's certainly comment well taken. I mean, yeah, we should look at the terpenoids and the alkaloids, but I'll say you got to start somewhere.
Thank you, Dr. Jonathan Cachat. Don't forget, next week we will be hosting a Reddit AMA with Dr. Cachat at reddit.com/r/kratom. Go there now. Maybe the announcement's already up. Maybe it's not. But it'll be next week. The music is Moonrunner by Captain Bigwheel. The Kratom Science podcast is produced by me, Brian Gallagher, for KratomScience.com. Take care.